Hello everyone, welcome to our second lecture of chapter 11. Today we're going to be looking at how muscles actually contract. We're going to be looking at the changes that occur inside of a muscle cell that actually allow for and cause a contraction to occur. So let's start with our not really attendance questions for the day. First, what is the difference between a neuron and a nerve? And second, a muscle is innervated by how many nerves? So go ahead and pause the video and try to answer those. And now let's see what the answer to those questions actually is. First, what is the difference between a neuron and a nerve? Well, remember, a neuron is a nerve cell. But a nerve is actually a bundle of axons. So if I have a cell, well, there's a neuron and then it's got its axon. But if there was another neuron nearby, it may have an axon going the same direction and another neuron nearby with an axon going the same direction. Well, these axons may be collected in some connective tissue, and this would be a nerve. So a nerve is just a bundle of axons. Specifically, it's a bundle of axons in the peripheral nervous system. Next, a muscle is innervated by how many nerves? Well, a muscle is the whole organ. Remember that from the self-study part of chapter 10 and 11. And the one muscle actually has many muscle cells inside of it. And one nerve supplies a muscle. But since there are a lot of neurons inside of a nerve, as we see in the first question, we can have different neurons supplying different muscle cells. But the muscle as a whole is innervated by a single nerve. So, just to kind of refresh before we dive into new stuff, this came from part of the self-study. But now that we've talked about most of these items, let's go back and see the hierarchy of a muscle, or the parts that make up a muscle, and everything that we've been talking about so far. So here is a muscle that has been cut in cross-section so that we can see inside of it. Remember, a muscle is an organ. It's the whole structure, like, for example, your biceps brachii. Here we can see that it's connected to a bone by way of a tendon, but we're concerned with the muscle itself, which has been cut. And inside this muscle, when we cut it, we can see that there are different little packets of structures inside. Well, each of these little packets is called a fascicle. And we've pulled one fascicle out right here, which is what we see in the next level. But before we go down to the next level, let's look at some of the membranes, the connective tissue sheaths that we can see here. Well, the muscle itself is wrapped by a connective tissue called the epimysium. The epimysium is continuous with the tendon, if there is one. The tendon then is continuous with the connective tissue that wraps the bone. Remember the periosteum? Well, the epimysium and the periosteum are continuous with each other. There may be a tendon between them. But that's how a muscle is anchored to a bone. But let's go back to our fascicle. We've got a fascicle here, and we've isolated one, so now we're looking at it. The fascicle is surrounded by a connective tissue called the perimysium. And here we see the fascicle also contains smaller units still inside, and we've pulled one of them forward. This is called a muscle fiber, which we learned is also called a muscle cell or a myocyte. So we've pulled a myocyte or a muscle fiber out, and that's what we're looking at down here. And the muscle cell is, we don't actually see it in this picture, but there's an endomysium wrapping the muscle cell. Just underneath the endomysium is the plasma membrane of the muscle cell, the sarcolemma. If we peel back the sarcolemma, we can see that there are smaller units still inside this muscle cell. These are the myofibrils. So here we've isolated a myofibril. We can see the sarcoplasmic reticulum, the T-tubule, kind of running around that myofibril. The myofibril, you guessed it, contains smaller structures still, and we've pulled some out. These are the thick and thin filaments. So we've pulled a myofilament out here, and we can see now we're at the unit of the sarcomere. So the sarcomere actually makes up the myofilaments we see down here, which is what's pulled forward here. So going the other direction, myofilaments are made of sarcomeres, end to end to end. But myofilaments collected together make a myofibril. Myofibrils collected together are inside of a muscle fiber, which is kind of surrounded by its plasma membrane, the sarcolemma, surrounded by an endomysium. Many muscle fibers together, collected as a fascicle, surrounded by a perimysium, and many fascicles together make a muscle, the organ, surrounded by an epimysium. So we can see each one of these units is just collected together to make the next level up. Now, real quick, something that I didn't mention last time because it's actually going to be much more important today, but when we were talking about the different proteins that make up the myofilaments, we talked about actin. And actin had that active site, but the active site was covered up by tropomyosin, which was held in place by troponin. Hopefully this is all sounding familiar. And then the thick filament, shown down here in purple, the myosin, there were these heads that kind of stuck off from it in all directions. Well, that active site on actin really is attracted with myosin heads. Myosin heads and the active site on actin really like each other. And the myosin head wants to try to attach to the active site on the actin. So real quick, let me draw. Here's a myosin. I'm going to draw one myosin head coming off. And remember, there would be lots of those myosin heads sticking off. And then there's the fibrous actin made up of G-actin subunits. And each of those actins has an active site. But the active sites are covered up by tropomyosin. Well, if something were to happen to that tropomyosin and it were destroyed or removed or something, this myosin head would actually raise up. So I'm going to draw another myosin head just so you can see what I'm talking about. It would raise up and attach to that active site. And when that happens, that is called a cross bridge. I may have mentioned this in the previous lecture, but I don't believe I went into a lot of detail. It's going to be very important today, which is why I wanted to talk about it in this lecture. A cross bridge is what happens when a myosin head attaches to the active site on actin. But that doesn't happen in resting muscle because we actually have that tropomyosin that's kind of wrapped around the actin in such a way that it covers up the active site. Okay, so again, this is all review so far. This is just to prep us for the things that we're going to talk about today. So here we see our myofilaments, and we see our sarcomere down here at the bottom. And we said that the sarcomere was the contractile unit of muscle. And when we think about muscle contracting, what's a good way to describe a muscle as it contracts? What does it do? It gets shorter, all right? So if, for example, this is a resting muscle, the whole organ, when it contracts, it's more like this. It gets shorter. So when we look at the proteins in a sarcomere, the actin and the myosin especially, it's tempting to think that during a contraction they probably get shorter. But actually that's not the case. Muscles contract using something called the sliding filament theory. And the sliding filament theory says a lot of things. But the main thing that it says is that during a muscle contraction, the proteins involved, especially the actin and the myosin, do not get any shorter. Instead, they slide past each other. I'll say that again. During a muscle contraction, the proteins involved, and when we're talking about the proteins, we're really mostly talking about the actin and the myosin, they do not get shorter. Instead, they slide past each other. So, what do I mean by that? Let's look down here at this picture. This muscle is actually pretty contracted. If we looked at this actin when this muscle was completely relaxed, the actin would actually end about here. Let me put a mark there so that you can see. And during a contraction, the actin would be moved towards the midline. And the same thing over here. The actin would end about right there, and it would be moved towards the midline. So during a contraction, the thin filaments of each of these sarcomeres is going to be pulled towards the M line of that sarcomere. And when that happens, the actin, when that is being pulled towards the M line, the I bands shorten. The I bands shorten. Also, the H zones shorten. Now, depending on how much of a contraction we're getting, the H zone might completely disappear as this actin is pulled into the M line. So the H zone is going to shorten. The H zone might disappear. The distance between the Z discs is going to shorten. So basically the sarcomere itself is going to shorten. The A bands, so all of this area right here, and all of this area right here, and all of this area right here, the A bands are going to get moved closer together. Why? Well, because we said the I band is going to shorten. If the I band shortens, then the distance between the A bands gets smaller, so the A bands move closer together. Now, each of these sarcomeres, remember, end to end to end, make a myofibril. And the myofibrils, remember, are held to the endomysium by those proteins like dystrophin. So, as the sarcomeres shorten, the myofibrils shorten. If the myofibrils shorten, since they're anchored to this endomysium, it makes the entire cell shorten. And if the cells are the length of a muscle, as they shorten, the muscle is going to shorten. So that's kind of at the big scale how a muscle shortens using the sliding filament theory. But that still doesn't tell us what's going on to cause this sliding filament to happen. And that's what the rest of today's lecture is going to look at. We're going to look at all of the details that are going on inside of a muscle cell that cause these contractions to happen. So let's learn some terms. Let's talk about motor neurons, let's talk about motor units, and let's talk about somatic motor fibers. Well, somatic motor fibers are axons of neurons from the central nervous system. So the cell bodies are in the central nervous system. The axons are going to extend from the central nervous system towards the muscle cells. Well, those axons are called somatic motor fibers. Somatic motor fibers, axons of neurons whose cell bodies are in the central nervous system. And these axons are going towards the muscle cells or the muscle fibers. So what are motor neurons? Well, motor neurons are neurons that innervate skeletal muscle. Now, when a nerve, which we learned at the start of this lecture, when a nerve goes to a muscle, the nerve actually has the neurons inside of it branch out, and different neurons will connect with or synapse with different muscle cells within that muscle. Here it shows us 2 of them. In reality, there would be many, many, many of them. Now, if we look, these motor neurons, well, they start to branch out at the end, near the axon terminals. Remember the terminal arborization that we talked about in the last chapter? Well, the motor neurons at the axon terminals are going to start to branch, and different branches will go to different muscle cells. Sometimes it's a whole lot. Sometimes it's just a few. But also, we'll have another motor neuron that branches and goes to different muscle cells. So one motor neuron can actually innervate and does innervate multiple muscle cells. But one muscle cell is only innervated by a single motor neuron. Let me say that again. One motor neuron will innervate multiple muscle cells, but one muscle cell is only innervated by a single motor neuron. So let's demonstrate that. Let's look at this purple motor neuron. It branches, and we can see that it innervates this muscle cell. I'm going to highlight it. It innervates this muscle cell and this muscle cell. So the purple innervates 2 muscle cells. Now, if we look at this muscle cell, it's only innervated by the purple. It's not innervated by the purple and the red. Same thing down here. This one is only innervated by the purple. It's not innervated by the purple and the red. The same thing is over here with this kind of pinkish red motor neuron. It innervates 3 muscle cells, but each of those 3 muscle cells is only innervated by the pinkish red motor neuron. So what's the point of that? Well, this is what we call a motor unit. A motor unit. A motor unit is described as a motor neuron and all of the myocytes that it innervates. A motor unit is a motor neuron and all of the myocytes that it innervates. So we see two motor units in this picture. The purple one, so that's this motor neuron and these two muscle cells. The pinkish red one is this motor neuron and these three muscle cells. And we can talk about motor neurons being large, in which case there are lots of muscle cells innervated by a single neuron. Or small, in which case one motor neuron innervates just a few muscle cells. Why would we need any difference? Well, large motor units tend to be found in areas where we need strength. Small motor units tend to be found in areas where we need very fine motor control. Now, what happens? What's the difference in a motor unit? Well, if we send an action potential through purple neuron, well, it's going to cause the two muscle cells that it innervates to contract, and none of the other muscle cells will contract, only these two. If instead we send an action potential through the pinkish red motor neuron, then the three muscle cells that it connects to will contract, and none of the other muscle cells will. We could send action potentials through both of these neurons, in which case all five of these muscle cells would contract. Okay, so now we're seeing different neurons will synapse with different muscle cells. And when a neuron sends an action potential to a muscle cell, ultimately we get a contraction in that muscle cell. So here we also see an area where motor neurons, the black fibrous guys and these large black ovals, are connecting with or synapsing with muscle cells, these pink stripy guys here. So each place where a motor neuron ends, we have this terminal button, terminal bouton, and it forms a synapse with a muscle cell. Now, this is just a synapse, but we're going to give it a special name. We're going to call this a neuromuscular junction. A neuromuscular junction is just a type of synapse. It's a synapse between a neuron and a muscle cell. Now, the part of the neuron here at the end, remember we talked about that already, that's called a terminal bouton or a terminal button. Then we have the synaptic cleft, and the part of the muscle cell that it is synapsing with is called a motor end plate. A motor end plate. And the things that we learned about at the last lecture of the nervous tissue lecture, the exact same things are going to happen here. An action potential is going to travel down the axon. It gets to the terminal button. It opens voltage-gated calcium channels. Calcium rushes in. Calcium binds to vesicles. Those vesicles release acetylcholine, and acetylcholine travels across the synapse, the synaptic cleft, and binds to those ligand-gated channels. Once that happens, we're going to get a voltage change in the muscle. So this looks just like the action potential that we saw in the previous chapter. But there are some very subtle differences. In a muscle cell, resting potential is actually negative 90 millivolts. Negative 90 millivolts. Remember, in neurons, it was negative 70 millivolts. Remember, we had said that every cell in our body is negative on the inside compared to the outside at rest, and different cell types have different voltages? Well, now we're seeing that neurons, negative 70. Myocytes, negative 90. But there's another difference that's a little harder to see, and that's over here. Over here in a neuron, we would get hyperpolarization. It would go down and back up. That doesn't happen in a myocyte. Muscle cells do not hyperpolarize. Neurons do. So, like we just said, those ligand-gated channels open. But what happens? Here it's a little bit different also than in a neuron. In a muscle cell, it's still an acetylcholine ligand-gated channel. But this ligand-gated channel, when it opens, two things are going to pass through it. Sodium is going to rush into the cell. Potassium is going to rush out of the cell at the same time. We've got sodium coming in and potassium going out. Now, it seems like that should make no difference because we've got positives coming in, we've got positives going out. They should be equal, right? Well, remember when we were talking about gradients? Sodium had an electrical and concentration gradient in the same direction. They were both inward. Potassium had an electrical gradient holding it in and a concentration gradient outward. So, more sodium comes in at this point. More sodium comes in and less potassium goes out. So we have more positives coming in than going out. And what happens is right here, right at the synapse, we get a little bit of a change in voltage. This part of the membrane is going to become less negative, more positive. And this is called an end plate potential. An end plate potential is the change in voltage at the motor end plate. All right, so now during this, there's going to be a lot that we talk about today. And I'm going to break it into 3 different areas. And we're going to watch a video after we talk about each of the areas. I'm going to talk over the video this time because I'm fairly certain that you guys don't hear the audio. Now, like always, this video is in your PowerPoint, so please watch the video with the original audio. But in this lecture, I'm going to talk over the video so that we can talk about it. This first part that we've talked about is excitation. We are looking at how a neuron stimulates or excites a muscle cell. And that's going to be the first video here that we watch. Okay. So here's the nerve going to the muscle. Now we can see the different neurons branching out. Here's one neuron showing one neuron to, well, the whole thing is one neuron. Looking at one neuron synapsing, here's the motor end plate. So now we're at the synapse. Here's our terminal button. Here's our motor end plate. There's the action potential arriving at the terminal button. Now we're seeing the vesicles inside that contain ACH. Here's our voltage-gated calcium channels. When the action potential arrives, they open. Calcium comes in, binds to the vesicles. Now they're exocytosing the acetylcholine into the synapse. There's our green ligand-gated channels. The black balls are the acetylcholine. The ligand-gated channels open. Now sodium in red is rushing in, potassium in blue is rushing out. Look to see there's a lot more red coming in, not as much blue going out. This membrane is becoming less negative, more positive. So we've just depolarized this part of the muscle fiber. And now it's just showing how does that signal end. Just like in a neuron, we're going to have acetylcholinesterase come along. So we've just excited that muscle cell. Now, everything that we just talked about has happened. We've just excited, we've caused an end plate potential here, and remember all of those proteins that we talked about when we were doing action potentials in a neuron? Well, those exact same proteins are all along the sarcolemma. And an action potential is going to spread in all directions across the sarcolemma, exactly the same way that it did in a neuron. Now, we get to a point where there's a T-tubule. Well, remember, T-tubules, all they are just enfoldings of the plasma membrane that run through the muscle cell. So, they have all of those same proteins. When an action potential is traveling along the sarcolemma, it also travels down the T-tubule. So here it's traveling along the sarcolemma, it's going to travel down the T-tubule. But inside the T-tubule, remember, we have the sarcoplasmic reticulum, the terminal cisternae of the sarcoplasmic reticulum, held to the T-tubule to make a triad. Okay, so now this action potential is traveling down the T-tubule, and all of those same proteins are there until we get to a triad. And now there's a new type of protein. It's something called a ryanodine receptor. That's what we see here, these purple guys. These purple guys anchor the terminal cisternae to the T-tubule. But they are also voltage-gated calcium channels of a new type. And when an action potential passes over those ryanodine receptors, they change shape. And they change shape so that there is a pathway from the inside of the sarcoplasmic reticulum through the ryanodine receptor, not out into the T-tubule, but instead out into the sarcoplasm of the muscle cell. So now we have just released a lot of calcium from the SR into the interior of the muscle cell. And remember what we keep saying over and over this semester, about calcium. If it's inside of a cell, we keep it packed away in the smooth endoplasmic reticulum because it likes to bind to things. And that's exactly what it does. Calcium, one of the things that it likes to bind to is troponin. And remember, the job of troponin is to hold the tropomyosin in place. But troponin is a protein. Think way back to near the beginning of the semester. Anytime we add something to or take something away from a protein, what will it do? It will change shape. So calcium binds to the troponin. We've just added something to it. It's going to change shape. Well, since it's anchored to the tropomyosin, when the troponin changes shape, it pulls the tropomyosin out of the way. That exposes the active sites on the actin. And once exposed, what did we learn? Myosin heads really like those active sites. And now that those active sites are exposed, what will we get? We'll get a cross bridge. The myosin head will reach up and attach to the active site on the actin. We're about to get a muscle contraction. We're not quite there yet. This is the part that's actually going to move from that excitation that we just saw and lead to the contraction that we're about to see. So this part is called excitation-contraction coupling. Now we're going to watch this video. So here's our muscle cell. Here's our T-tubule. And here's all of the sarcoplasmic reticulum. There's the SR. There's our triad, T-tubule, terminal cisternae, terminal cisternae. There's our ryanodine receptor. Lots of calcium inside the SR. There's our action potential. Now calcium is flooding into the interior of the muscle cell. I apologize. I apologize. Okay, I apologize for that. There was some sort of glitch with my computer and it wouldn't respond. Hopefully, you were able to hear everything and can still hear me. So, that calcium rushed in, it bound to the troponin or troponin, and that pulled the tropomyosin out of the way. We formed a cross bridge. Now we're going to get to see what happens as a result. So at rest, let me back up here. At rest, the myosin heads are not attached to actin because the active sites are covered up. Now, even though we don't see it in this picture, I'm going to draw something in. We've got ADP and inorganic phosphate. And those are both attached to the myosin head. But the moment that the myosin head attaches to actin, that's going to knock off the inorganic phosphate. As soon as we form a cross bridge, we're going to knock the inorganic phosphate off. And once the inorganic phosphate falls off, this bond between actin and myosin gets stronger. And when the bond between actin and myosin gets stronger, it also knocks the ADP off. Here in this image, it shows them both happening at the same time. In your book, I believe it also shows them both happening at the same time. But in reality, when the cross bridge forms, inorganic phosphate falls off, makes this bond stronger, which knocks ADP off. So now we've removed something from this protein. What happens when we remove something from a protein? It changes shape. What kind of shape change do we get? Well, the myosin is going to bend a little bit. It's going to bend towards the M line. So real quick, let me draw out just a thick filament. We've got a bunch of myosin heads down here. We've got a bunch of myosin heads down here. Well, these down here would bend this way. These down here would bend this way. So we get a bending. But since the myosin heads are attached to the thin filaments, I'm going to draw a thin filament as a dotted line here, and a thin filament as a dotted line here. If the myosin heads are attached and it bends forward, it's going to push the thin filament this way, towards the M line. Same thing over here. If this myosin head is attached and it bends forward, it's going to push this thin filament towards the M line. What's going to happen to the Z discs? They are also going to get pushed. So we've just shortened the sarcomere a little bit. This is called the power stroke. Well, when this myosin head bends forward during the power stroke, that's actually going to cause ATP to really, really like myosin. When myosin is in this bent forward position, ATP really likes it. So ATP comes along. And when ATP attaches, well, now we've added something to a protein. It's going to cause the myosin to change shape. It's going to break the cross bridge, and myosin bends back down. It pulls away from the actin. Well, that requires energy in the form of ATP. So ATP is needed to break the cross bridge. Remember, myosin really liked actin? ATP is needed to make myosin pull away from actin. Once we break the cross bridge, it's going to hydrolyze or split the ATP. When the ATP is split, we get the same thing we always get when we hydrolyze ATP. What's that? ADP and phosphate. That's where that ADP and phosphate originally came from. Now, As long as a motor neuron is still sending action potentials, then calcium will still be present inside of a cell. And if calcium is still present inside this muscle cell, then calcium is still attached to the troponin, which means the tropomyosin is pulled out of the way and the active sites are still exposed. If that's the case, then we continue to go through this. So we get formation of a cross bridge, knocks the inorganic phosphate off. Stronger bond, knocks the ADP off. We get a power stroke. Once we get the power stroke, ATP really likes the myosin. It attaches, it breaks the cross bridge. ATP is hydrolyzed, it's split into ADP and inorganic phosphate. The hydrolysis of ATP, causes the myosin to bend back to its original shape. We say it cocks it into the high energy position. All that means is just the head raises up and it's ready to go again. And as long as calcium is present, we keep going through this. And each time, the sarcomere is shortened a little bit more, and a little bit more, and a little bit more. One single cycle does not shorten the sarcomere very much. And one single cycle doesn't shorten the muscle detectably at all. But luckily, when we're contracting a muscle, say when you go to pick something up and your biceps brachii shortens, that's not the result of an action potential. That's the result of multiple action potentials. So this is called the cross bridge cycle. Let's watch this. There's a little bit of an overlap here. Here it's just kind of showing it, and then in a minute it's going to go back and do it in detail. There's our contraction. Myosin head with, there's our thin filament, actin, tropomyosin, troponin. There's our ADP and inorganic phosphate. They've just formed our cross bridge. So now we're going to do it step by step. We're seeing where the ATP comes along. So there's our ADP and inorganic phosphate. We're seeing where they came from and why they were there in the beginning. So here we go. We've got our calcium on already. The tropomyosin's pulled out of the way. Just formed the cross bridge. Knocks off the inorganic phosphate. Strengthening the bond. ADP is knocked off. Myosin bends forward, power stroke. Have to break the cross bridge. How do we do that? There's our ATP coming along. That breaks the cross bridge. Hydrolyze the ADP or ATP into ADP and inorganic phosphate. Cock it into the high energy position. Now it's ready to go again as long as calcium's still present. And we can see that lots of myosin heads on one thick filament are all working together to move those thin filaments. Here's the H zone. We're going to see it shorten. And eventually, now it's disappeared. That's what we were talking about earlier. But when a muscle's relaxed, that active site's covered up. So that's how a contraction happens. But what about when we need the muscle to relax again? Well, once we're done contracting a muscle, we stop stimulating it. We stop sending action potentials. So if there's no more action potentials traveling along the sarcolemma, there's no more action potentials traveling down the T-tubule. That causes those ryanodine receptors to close. That means calcium stops coming out of the sarcoplasmic reticulum. But there's still calcium in the sarcoplasm. There's still calcium inside the muscle cell. Well, inside the sarcoplasmic reticulum, there's a protein called calsequestrin, and it really, really, really likes calcium. So calsequestrin kind of acts like a vacuum. It sucks the calcium back into the sarcoplasmic reticulum, and then it holds on to it inside. Now we've got calcium has been removed from the inside of the muscle cell. It's pulled away from the troponin. So the troponin goes back to its original shape, which means tropomyosin goes back to its original position, covering up those active sites. And remember at the end of the sarcomeres, there was that little spring-shaped protein, titin? Titin acts like a spring. It pulls everything back to its original starting position. The sarcomere snaps back to its resting state. Once that happens, the muscle's relaxed again. But let's talk about something else weird that happens with muscle. Let's talk about rigor mortis, which is contraction of muscle after death. And that's what we see right here. This person obviously very dead. He does not have the top of his head. Looks like he doesn't have a brain inside right now. But what's happening here? The muscles in his back are contracting. Well, he's not telling them to contract. He doesn't have a brain to tell them to contract, but they're still contracting. So what's happening in the case of rigor mortis? Well, first let's go back and think about everything that was happening during the cross-bridge cycle. None of that required life. Everything that was happening there was a chemical reaction. Everything in the cross-bridge cycle is a chemical reaction. So, what happens after death? When someone dies, they stop stimulating. They completely go relaxed. I believe by now you've already had your muscle lab, and you've talked about muscle tone, and that is that we are constantly stimulating our muscles. There's always a low level of tension in our muscles. Even when you are your most relaxed, there's still a little bit of tension there. But once someone dies, they stop sending any action potentials to their muscles. And we go completely relaxed. It feels very different. If you've ever picked someone up while they were asleep, well, they still have some muscle tone. If you've ever had the unfortunate need to move a dead person or a dead animal, you know that it feels very different from when they are asleep. They are very, very soft, very relaxed. There's no tension in the muscle at all. And it stays that way for quite a while. And it will change depending on temperature and other environmental factors. But after a certain amount of time has passed, we're going to start to get breakdown of membranes, including the plasma membrane of the cell, including the endoplasmic or sarcoplasmic reticulum. So what happens is calcium starts to leak into cells, especially muscle cells in this instance. And when calcium leaks into a muscle cell, what's going to happen? It's going to attach to things. It's going to, if we're talking muscle cells, it's going to attach to troponin. What happens when calcium attaches? It's going to change shape. When it changes shape, it pulls tropomyosin out of the way, exposing the active sites. This doesn't require life. These are chemical reactions. When that active site is exposed, what happens? Myosin really likes that active site, so it's going to attach. And we go through the cross-bridge cycle. And we shorten the muscle, and that happens over and over and over, and the muscle contracts and contracts and contracts. And that keeps happening until something else. What is it that breaks the cross bridge? ATP. Well, we're going to keep using up that ATP to break those cross bridges and keep going cycle, 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 shortening the muscle even more. Eventually, we're going to use up the ATP. ATP does require life. We have to have cellular respiration to make ATP. And if we're dead, we've got a limited amount of ATP. We go through those cross-bridge cycles, shortening the muscle, shortening the muscle. Eventually, we run out of ATP, and we can't break the cross-bridge. So we go through a contraction, and then we're stuck midway through, and we can't break those cross-bridges. And that's what we see right here. That's rigor mortis. And it will stay that way, again, depending on environmental conditions for quite a while, until even more decay happens. Eventually, we start to break down the proteins, the actin, the myosin, other proteins in the body. Once they start to break down, then we go back to a relaxed position. So, rigor mortis is the result of depletion of ATP after death. So we can't break the cross bridges. And forensics can look at things like the environment, the temperature, things like that to see, okay, has this person started rigor mortis yet? Are they still in rigor mortis? Or has rigor mortis passed? And based on that and environmental conditions, they can gauge approximately how long it's been since somebody has died. Okay, so that's where we're going to stop today. Our next lecture, which is our final lecture over muscles, we'll be looking at factors that influence contractile strength. All right, so if you have any questions at all, please let me know. I know this was one of our tougher lectures, so watch it several times. Watch those videos that are in the PowerPoint. Ask questions if you have them. Otherwise, I'll talk to you next time. Take care.